speaker, Nilika Malavige is a professor and head of the Department of Immunology and Molecular Medicine at University of Sri Jayawardhanapura and an academic visitor at the MRC Human Immunology Unit, University of Oxford. She is also a member of executive committee of the International Society of Infectious Diseases. She was also recently appointed to the WHO Technical Advisory Group on COVID-19 Technology Access Pool. In addition to that, she has published more than 100 peer-reviewed journals in international peer-reviewed journals. Then I would like to invite Professor Nilika to start her presentation.
Thank you so much, Samita Kamarkom. Thank you for the invitation. Now I can. Okay. So hope everybody can see my screen. I'll just get my. Okay, so can everybody see my screen? I hope you can. So I think I'll start with the current situation where we are, and then talk about what we can expect with these vaccines and variants. Now, these are just two figures. One is a cartoon and the other one is the cover page of Newsweek. Now, as we know, right now, so many countries in the world are seeing a very high caseload of COVID again in 2021. The year we thought that we would go back to normal turned out to be worse than 2020 actually. So despite in the 8th month of 2021, we are seeing even more devastating effects due to COVID because of this emergence of the variants. And the Delta variant is, of course, named by Newsweek magazine as Doomsday variant. Now, whether it, let's discuss to see whether it's actually so bad as it is claimed. And of course, in Sri Lanka, this is a recent cartoon which was out in a newspaper, where the doctors seem to be magnifying appearing to be magnifying the actual situation, whereas the politicians seem to be, looking at the other end of the telescope and finding it as a really small problem. So how bad is the problem? Now, this is, I hope you can see these graphs. I mean, you can access them anytime from our world in data. This shows the number of cases per million, because you can't go by the total cases because the population in each country is different. So, but if you go as the number of cases per 1 million population, Malaysia is seeing a really high number of cases right now. UK saw a very high peak of cases and it's sort of in 400 cases per 1 million people. And of course, Sri Lanka is way far below than these countries I have chosen to highlight as it comes to cases per 1 million. But what about deaths per 1 million people, individuals? So we know that Malaysia and Indonesia are having a really bad time. So they have 6 deaths per million people. And Sri Lanka is currently having 4 deaths per million people. And although there was a very high caseload in the UK and United States, the deaths per million cases are very much lower than what you see in Malaysia, Indonesia, and Sri Lanka. And so the question is, why do some countries have low death rates compared to others? Is it because UK and USA have different variants? Maybe they don't have Delta. Or is it an effect of vaccination? Now, if we come to there, and this is actually, so how, now when we say that Sri Lanka has this many cases, what is the positivity rate? The number of cases actually depend on the number of tests that you carry out. So in Indonesia, the positivity rates are close to 25%, which means they're actually grossly underestimating the number of cases. Sri Lanka positivity rates are around 18%. Whereas in the United Kingdom, USA, although they have a very large number of cases, even India, India doesn't have a large number right now, although UK and USA have a large number of cases, their positivity rate is very low because they are doing a large number of tests and they're picking up a lot of patients. And so this just shows, again, the positivity rate in different countries. So Sri Lanka is seeing around 20% and is colored like that. So all these countries, although they are seeing high number of cases, even India, the positivity rate is less because the number of tests are more. And in certain countries like in Africa and Southeast Asia, the positivity rate is more. And and how much testing are people doing per 1,000 people? Again, because it depends on the population, you can't just say this number of tests. And the Western countries, because they have more facilities, they have more money to invest on testing, they are doing lots of tests. So they are doing quite a large number of tests per 1,000 people. India is doing more than Sri Lanka right now. Sri Lanka is doing about 0.1 to 1 test per 1,000 people. And we have to take all this into account when we talk about our case loads and tests. Now, the other one is contact tracing. So how many tests do you conduct for newly confirmed patient? In Sri Lanka, it is not that many tests conducted per new identified case, whereas in other countries, again, this is the difference between the wealth that different countries have. We know that Sri Lanka is going through economic crisis as well. So our investment on testing has not been that great for reasons that we understand. Now, if we look at the Sri Lankan situation, the rapid rise in the number of cases, there is a rapid rise, but and a very high positivity rate. And when you put both together, the rapid rise, high positivity rate, but low test numbers, the actual number of cases are actually a grossly underestimate of the actual number of cases. And we are seeing a rapid rise in the death rates, which are similar to seen in Indonesia and Malaysia. And so that is probably a better reflection of the actual number of people infected. But the other important thing that I would like to say here, which is quite important, is that when somebody gets infected, they take a few days to show symptoms. And after showing symptoms, it takes like 7 to 10 days to develop severe disease manifestations, and then a few more days to die. So basically now, for instance, we had 1,011 deaths yesterday, and those 111 deaths yesterday, and those 111 deaths yesterday is a reflection of an infection that happened 2 weeks ago. Okay, because so the deaths we are seeing right now reflects the situation 2 weeks ago and not the situation right now. So the deaths due to the situation right now will be seen in 10 to 14 days. This is because the number of deaths lag behind the infection, because it takes time to get infected, show symptoms, then severe disease and die. So apart from these deaths, so those are the confirmed deaths. So it's important to find out how many deaths happen, because for instance, in India, although the actual number of deaths that they had declared is like 400,000, the excess deaths, when you look at the number of people who actually died in those given months compared to previous years, they estimate like 4 million people died in India instead of 400,000, which is 10 times more people died during the main Delta epidemic in India than they actually declared because of reporting issues probably. So now, coming to all that is our higher mortality rates. Now, I just showed you that although in the UK and in the USA, although they have higher number of cases, the mortality rates are less, and India had high mortality rates, which were not shown in their figures, and we have higher mortality rates, and is it because we are having a Delta issue? Now, this is the SARS-CoV-2 variants found in different countries. So in Bangladesh, it's 100% Delta is seen. In the United Kingdom also, the high number of cases are because of Delta. The Delta is 100% in USA, Indonesia about 93%. So, I mean, we haven't got there yet as far as Delta is concerned. So as of now, about 44% of, this is based on sequencing data, 44% of infections are by Delta, but it depends on where we are testing. So I'll come to that. So when it comes to vaccines and variants, the questions that we actually need to answer, or I think everybody wants to know, do vaccines prevent symptomatic infection from variants? Okay, is preventing symptomatic, can we achieve that? And of course, do vaccines prevent death? So if you get a vaccine, does it mean that you're not going to die? Why do some people who have both vaccine doses still die? I mean, there are certain media reports showing that certain people, even after taking both doses, still do die. And what does that mean? And can you transmit infection after being fully vaccinated? Okay, because we know in a particular time that the vaccines were given to between the ages of 30 to 60-year-olds, the working population in Colombo, with thinking that they will stop transmission and taking the infection back home. So are vaccinated individuals, do they not transmit infection? And of course, all vaccines have a similar efficacy. Now, coming to all that, I'll just tell you about what sort of antibodies or immune response you need to get protected. So now this is the SARS-CoV-2 virus. It is called a coronavirus because of a crown-like appearance, and it has the spike protein jutting out. So this is how the spike protein looks like, and the spike protein binds to the ACE2 receptor. So this binding of the spike protein to the ACE2 receptor is by this receptor binding domain, and it is antibodies against this particular area that is most important in preventing infection. So, but when somebody has been infected with a virus or been given a vaccine, depending on whether it's inactivated vaccine, then you develop antibodies to the whole viral particles that are accessible. And if you're given a vaccine with just the spike protein, you develop antibodies to various parts of the spike protein. So you develop antibodies to the receptor binding domain, the intraimmunal domain, the S2 subunit. But just so the mere presence of antibodies alone is not a reflection of how protected you are. So because as I showed you earlier, the virus attaches to the S2 receptor by the receptor binding domain. So that antibodies that bind to the receptor binding domain, specifically the areas that are most important to bind the S2 receptor, are the neutralizing antibodies. So it's the presence of neutralizing antibodies that are associated with protection, that has been shown to associate with protection, rather than just the presence of IgG antibodies. Now, so this is just the cartoon, what the SARS-CoV-2 protein looks like. And so the spike protein is coded to generate the folded spike protein. And these variants, so-called variants, acquire mutations specifically in this receptor binding domain, so that it can bind to the ACE2 receptor with a higher affinity, or when they change the protein, the binding of neutralizing antibodies is not that great. So by this changing of these various positions, so then when the nucleotide changes, the amino acid changes, and because when there's a change in the amino acid, it increases the binding. So when the binding is increased, that means the cells can be infected with a lower viral load, so the transmissibility is higher. And when these mutations occur, certain mutations stop binding of neutralizing antibodies, and that would lead to, of course, escaping immunity. So now there are so many SARS-CoV-2 variants. Okay, I mean hundreds of variants based on the mutations with various names, various pangolin lineages. But there are the variants of concern are named because either the variants of concern are associated with an increase in transmission, increase in disease severity, escape from neutralizing antibodies that are generated because of previous infection or vaccination, or evade diagnosis by PCR. So when we look at the SARS-CoV-2 variants, the WHO has named 4 variants of concern. And so because of those reasons. So one is this alpha variant, which was previously called the UK variant. It was the dominant variant in all over the world, affecting 182 countries until, of course, Delta came. So there is a beta variant, which is the South African variant, which is very good at evading immunity. The gamma variant, which is not so prevalent in as other variants, but so initially we only had alpha, beta, and gamma, but then Delta emerged. So because Delta is so much more transmissible than other ones, it very soon replaced other variants in the affected countries. So now, how bad is Delta? Okay, so Delta is 40 to 60% more transmissible than alpha. So when we compare alpha, which is the UK variant, to the original or the ancestral SARS-CoV-2 virus, the alpha variant is 50% more transmissible than the original one. So, and delta is again around 50% more transmissible than alpha. So because of that, you can imagine the transmissibility of delta. And a recent study showed that the viral loads of delta is about 1,000 times higher than the ancestral SARS-CoV-2 variants. So because this is the Young Scientist Forum, I believe that people are quite familiar with molecular diagnostic assays and real-time PCR. So the cycle threshold values are really, really low with the delta, which is an indirect indicator of the viral loads. And we have got even CT values of 2, 3, 5 people with that low viral loads, high viral loads with the delta, which we didn't see initially. And as far as hospitalization is concerned, studies coming from Scotland do show that the hospitalization of unvaccinated individuals with delta was twice as more than unvaccinated individuals for alpha. So even with alpha, there were more people getting hospitalized than the previous variants, but with delta, it is likely to be more. Now, again, now when we look here about delta and even looking at Sri Lankan, the picture we are seeing in Sri Lanka and what we saw in India, and delta seems to be causing utter devastation, but whether it's because the rapid rise of number of cases exhausts all the hospital space available for care in hospitals, and that's why people die, or because people actually develop more severe disease, that is something that is currently under investigation. So this is just showing you how transmissible delta is compared to others. So, so the original SARS-CoV-2 variant, so this is if there are no measures in place, right? So the original SARS-CoV-2 variant basically infected around 3 people. And so to acquire herd immunity, I hope you know what herd immunity is. That is basically the proportion of people in the community who need to be immunized to stop transmission. So for the original variant, if we had immunized 67% of the total population, we would have stopped transmission. But the alpha came, which was more transmissible than the original one. So to actually limit transmission to a particular degree, then 75% of the population needs to be vaccinated. But then delta came. So delta is more transmissible than alpha. So on average, it infects around 6 people. So some of the studies actually show it's even more, infecting more than 6 people on average. So we really don't know about that. And the vaccinate for herd immunity, at least 80%, the minimum 83% should be immunized. This is fully vaccinated, not giving 1 dose. So that is a large number of population that needs to be immunized. And given that none of the vaccines have been tested in children under 12 years, so that no clinical trials have been done in children less than 12 years. In children 12 to 18 years, some vaccines are being given in certain countries, but in countries like the UK, although vaccines are available and the clinical trials have shown that it is okay to give children, they're still waiting for more data to see if vaccines are actually safe to be given to children. So that's a different story. Now, this is just showing when delta comes, what happens. So this pink is alpha. So this is data from the Public Health England. So this is UK data. So when alpha came, that became the dominant variant because it is 50% more transmissible than the previous one. So that was accounting for 100% infections. Then delta came sort of towards the end of March. And you can see how delta came and took over, completely displaced alpha within basically 2 months. Okay, just 8 weeks period, delta completely displaced alpha. And we know that in the UK completely fully opened in July. So during this time, from April to sort of June, there were restrictions in UK, not a complete lockdown, but certain restrictions. So despite that, because of the high transmissibility of delta, it completely replaced alpha. And this is our own data. So this is in the CMC Colombo. So by the first week of July, Delta accounted for like about 25% of infections. And by 31st of July, it accounted for over 90% of infections. This is in the Colombo Municipality Council area. This is by our variant PCR. And this is the sequencing data showing that we just had about 2% Delta by 9th of, so we initially detected Delta in the Colombo completely randomly. We just randomly picked it up during the second week of June, which doesn't mean that that's when Delta came into the community, because to detect something randomly in the community, and for that to be the first one you detect, the mathematical possibilities are absolutely, it is impossible. So although we detected it in second week of June, obviously that is not when Delta came into the community. And so these are the samples from all over the country, and Delta, as you can see, is taking over. But it is not everywhere. So we are so most of the Delta is in the Western province, and if you listen to Sri Lankan news, you would understand that that Delta is the Western province hospitals and Karapiti hospital that is facing the main crisis situation. And it just started emerging in sort of Kurunegala and Ratnapura and Jaffna, and whereas the hill country, Kandy, Matale, Nuwara, Badulla, Mahiyangane, all those areas still have alpha. Delta had not gone to these areas by end of July, which it will. I mean, it's absolutely guaranteed that Delta will go there and will spread all over the country. So this is just to show how things evolved in Sri Lanka. This is the initial viruses that were there in Sri Lanka, but then alpha emerged and of course displaced everything else, because alpha is 50% more transmissible than everything else. So, and of course, Delta is coming, and soon we will see how Delta takes over alpha and spreads all over the country. So coming to the question of how effective are these vaccines for Delta? So that is something that everybody is interested in, because I mean, it's large enough, and given that there is hardly any space in hospitals right now, nobody wants to fall sick with COVID. And we have several vaccines, starting from the mRNA technology, which was used first time in the world history for COVID. So we have Pfizer and Moderna. There are no other licensed vaccines. Then we have the viral vector vaccines. There are 4 types of viral vector vaccines. The first one that was used was a vaccine used for Ebola. So we have the AstraZeneca, CanSino Biological, which is Chinese vaccine, Gamaleya, which is Sputnik, and Johnson & Johnson. So the WHO has only approved the Oxford AstraZeneca vaccine and the Johnson & Johnson. The other 2 vaccines have not been approved by the WHO. Then we have whole virus vaccines, Sinovac, Bharat Biotech, the Indian vaccine, Sinopharm, and Medicogo vaccine. So of these, the WHO has only approved Sinopharm and later Sinovac. And then we have these protein subunit vaccines, and these have not been approved by the WHO. And we know that hepatitis B vaccine is a protein subunit vaccine. So how do these vaccines work? Now, Sri Lanka is doing really fantastic in vaccinating very fast. So as of 7th of August, we had fully immunized 13% of our population and given at least 1 dose to about 50% more. Whereas countries like the UK had fully vaccinated close to 60%, and the USA about 50%. Now, countries like Indonesia, Thailand, India, I mean, India is already fully vaccinated 8.2 right now, but when Delta hit India, which was end of April, basically April, India had only fully vaccinated 3%. So that caused a huge problem because Delta hit an unvaccinated population. So how does this sort of vaccination compare with the devastation that Delta can cause? Now, this is published data for the Pfizer and AstraZeneca. So this is just vaccine effectiveness as compared by symptomatic infection. So with 1 dose previously, so this is any vaccine. So if you take the Pfizer vaccine, the effectiveness for the alpha variant or UK variant with 1 dose was 47.5%, and it fell to 35.6% for Delta. Okay, so this is Pfizer. With one dose, you only have protection against symptomatic infection, 35.6%. But with both doses, even for Delta, symptomatic infection, there is 88%. Now, with the AstraZeneca Covishield vaccine, although it has had almost close to 50% efficacy with one dose against Alpha, the efficacy for Delta with just one dose is just 30%. But with both doses, it has significant efficacy against symptomatic disease. But it's not symptomatic disease we are worried about, but actually we are worried about severe disease and death. So how does that happen? So now this is, I'll just talk you through this graph. So this is again symptomatic infection, which I just showed. But what we are worried about is hospitalization. So for hospitalization, for Alpha and Delta, both vaccines, these are both vaccines together. The UK data showed good efficacy in preventing hospitalization. In other words, severe infection after both doses for Delta. So even with one dose, it significantly did prevent hospitalization, and with both doses, it's the 96% protection against hospitalization. So I know right now we are hearing that a lot of healthcare workers who have got both doses of the AstraZeneca or the Covishield vaccine are getting infected, but so far, not except for a very few handful of individuals, most of them are having asymptomatic infection or mild infection. So now, despite vaccine fully vaccinating about 60% of the population, we know that I just showed you that the numbers of infection in UK is increasing, but the number of hospitalizations have drastically decreased. So this was the wave that the UK saw end of December, early January, but this time the number of hospitalizations is very much less. So this is the projection without the vaccine, and this is projection with the vaccine. So although yes, people are getting hospitalized, but the numbers that are getting hospitalized are so much less, so that the hospitals are not overcrowded. The doctors and the healthcare workers have adequate resources to manage these patients. So that is a very good news. Now, this is our own data, which was published in Nature Communications recently. Just to show how much the AstraZeneca, the efficacy of AstraZeneca, not the efficacy, this is just the immunogenicity study, just to find out how many people develop antibodies. So here I'm going to talk about neutralizing antibodies, which we measured by surrogate viral neutralizing antibody assay, which is used for many vaccine trials. So, and you can see, so these are the blue dots are the people who did not have infection before vaccination. And after a single dose of AstraZeneca, 97.1% of individuals developed antibodies. I don't have two-dose data because we are still doing the analysis for the two doses, but this is just after a single dose of AstraZeneca or Covishield, 97.1% of individuals developed neutralizing antibodies. And in that study, we hadn't looked at Delta responses because we did the study in February, and during February, there was end of February, early March, there was no Delta threat, and we didn't have the assays to do Delta. But we recently took the serum and did the assays for Delta at 4 weeks, and again at 12 weeks. So we took the same people and fed them at 12 weeks when they got the second dose. And as you can see, at 4 weeks, vaccinees had significantly less responses to Delta than those with mild infection and moderate infection. So these are people who had natural infection. But after 12 weeks, those with natural infection also had a decline in the antibody levels to Delta. So although natural infection protects you against reinfection, again, the antibody levels sort of tend to drop with time. So at 12 weeks following natural infection, the antibody responses to Delta after a single dose of AstraZeneca and mild and moderate infection reduced. But and we also had the opportunity to look at 16 weeks because a large proportion of individuals in Sri Lanka did not get the second dose of the vaccine at 12 weeks. So then we looked at 16 weeks, and by 16 weeks, the neutralizing antibody proportion of people who had neutralizing antibodies had dropped to 76%. So there was a drop from 97.1 to 76% by 16 weeks for a single dose. And this drop was especially in older individuals. So everybody got a second dose, which is good because of this drop. And actually in countries like the UK, where the Delta emerged, not emerged, I mean, it's there now, they reduced the gap between the vaccine, the second dose of the vaccine, they brought it forward to 8 to 10 weeks because a single dose of a vaccine is less protective. And so I know we are using mainly using Sinopharm and Sputnik in Sri Lanka. I mean, Sputnik not that much. We were going to use Sputnik, but then we didn't get the opportunity. So Sinopharm is one of the main vaccines we are using in Sri Lanka. So we don't know the efficacy of Sinopharm for Delta, because to find out the efficacy or the effectiveness of a vaccine for any variant, we have to do a clinical trial. So there are no clinical trial data on or observational studies to look at the efficacy of any of these vaccines against Delta. And also there is no data on the effectiveness on reducing hospitalization and death with Delta. But what we do have is immunogenicity data. In other words, after receiving the Sinopharm vaccine, how many people develop antibodies. So this is available as a preprint at MedArchives, and you can see the seropositivity rates at 4 weeks and at 6 weeks. So the individuals between 20 to 40 years of age, after one dose, the seroconversion rate was 60.6%, which increased to 99% by 6 weeks. And but in the old age groups, the seroconversion rates were not that high as young individuals. So, and this was significantly less in those with older than 60. And but as mentioned, it's the neutralizing antibodies that are most important. So let me take you through this graph. So it's the blue dots that you need to look at. The red dots are those who were already infected at the time they got Sinopharm and got the vaccine. I mean, infected people are also encouraged to get the vaccine, but these infected people didn't know that they were infected by the time they got the vaccine. These studies were done at the CMC, and of course, we had enough people infected in the CMC by the time the vaccines were rolled out anyway. So these are the baseline uninfected individuals, and by 4 weeks, the neutralizing antibodies did increase, and by 6 weeks, they increased to levels seen in a similar level as following natural infection. So these are individuals who had natural infection. So after 2 doses of Sinopharm, 2 weeks after 2 doses of Sinopharm, they had similar neutralizing antibody levels as natural infection, and 81.25% individuals had neutralizing antibodies or S2 receptor blocking antibodies at 6 weeks. That is 2 weeks after the second dose. Infected individuals, of course, even after 1 dose, had very high levels of antibodies, and after both doses, they had extremely high levels of neutralizing antibodies compared to natural infection, which was seen with all vaccines. Now, again, we looked at the receptor binding antibodies to the receptor binding domain at 4 weeks. So this is after 1 dose of receiving Sinopharm. This is after 2 weeks after receiving the second dose, and this is for natural infection. So the green dots are for the original variant. So after the first dose, you don't develop that much of antibodies, but after the second week, you do second dose, you do develop quite a large proportion of antibodies, but the antibody levels are significantly less than naturally infected people for both alpha and the original variant. But for Delta, as you can see, even naturally infected people had less antibodies to Delta. So after 2 doses of Sinopharm, the antibody levels for Delta were similar to the levels seen after natural infection. So we don't know whether this means, what exactly this means, because this is immunogenicity data. But going by if antibody positivity, because the levels are similar to in natural infection as vaccination, maybe the people after 2 doses of Sinopharm may be having a similar protection as those with natural infection. But of course, we know that naturally infected people can get reinfected with variants. Now, the next question is, can people still die or get severe disease after being fully vaccinated? okay, because there were some deaths highlighted in media. So this is again data from the UK. So this is the second wave, which we saw from, like 8th of November to like February. And so these are the case numbers and cases per 100,000 population and deaths per population of 10 million population. So you can see a large number of deaths. This is the third wave that they're seeing right now. So these are the number of cases, but these are the number of deaths. So basically you can see, yes, some of the people are still dying, but vaccination has drastically reduced the proportion of people dying. But it is a relative thing. So basically the vaccines reduce the risk of dying due to COVID. So basically a person who is 80 years old with vaccination, the risk of dying significantly decreases. In other words, the same risk as an unvaccinated 50-year-old. Okay, so whereas if a 50-year-old is vaccinated, the risk of dying decreases to an unvaccinated, maybe 20-year-old. Okay, so this is why. So it is the old individuals who are dying, and this is precisely why it was recommended that old individuals, especially people over 60, be given the vaccine first, and then after the old individuals are fully vaccinated, you then move vaccinating young individuals. So basically, the plan for Sri Lanka, which was was exactly the plan that was carried out in all other countries, which is first vaccinate old individuals over 60s, then vaccinate people between the age of 30 to 60 who had comorbidities, because again, we know that it's people who have diabetes, heart disease, and so on who die. As of now, we know that so many people certain number of people are dying every day, and about over 75%, between 75 to 80% of people who are dying are people over 60 years of age. So this is why it was so important to vaccinate that group of individuals. And the next question is, of course, can fully vaccinated individuals transmit infection? So there have been 2 studies. So this is from the Public Health England. And so this is the viral loads in unvaccinated individuals and the viral loads in vaccinated individuals. So this was, so there was a bit of alpha then, but as Delta emerged and it was 100% Delta, you can see that there is no difference in the viral loads between people who are vaccinated and unvaccinated. And this is another study showing that at the beginning of an infection, both vaccinated and unvaccinated individuals have similar viral loads, but in people with who are vaccinated, the viral loads very drastically start to drop after 5 days, whereas the reduction in the viral loads is very much slower in people who are unvaccinated. So, can fully vaccinated individuals transmit infection? To answer that question, well, actually, the viral loads seem to be similar during early illness in vaccinated and unvaccinated individuals, but these studies looked at RNA or CT values and not actually infective viral particles. So, going by the viral loads as per RNA detection, they seem to be equally effective, but we don't know about the cultured virus in these respiratory secretions. But basically, it just looks like the vaccinated people are equally capable of transmitting infection. So this is my last slide because I think it's important to sort of answer questions and have a discussion. So will more variants emerge? That is a big question everybody wants to know. And if we find out, like last August, I think, a lot of people thought that the top was the SARS-CoV-2 is a very stable virus and we might not get new variants and so on. It was only a few people who thought by September that we might be getting variants. And of course, by December, it was very obvious that there are variants. And Delta emerged because of the high transmission rate that was seen in India. So when you give the virus the opportunity to transmit and replicate, you get more and more variants emerging. So will variants emerge? Yes, they will, given what is happening in South Asia and Southeast Asia. Because of this vaccine inequality, we know that all the developed countries have got a lot of vaccines, and they're talking about giving the third dose to their community, to their population. So by giving the third dose, although you might assume that they're being protected, because there's a ravaging outbreak in the South Africa, Southeast Asian and Asian region, there are variants are likely to emerge, and then the vaccinees won't be protected. So this is why it is, in order to be safe, it is important to vaccinate as many people as possible equally to reduce transmission. And the next question is, will everybody require booster doses? So some studies have emerged from Moderna, Pfizer, and even AstraZeneca, showing that immunity does last after vaccination for some time, even up to 8 months, good immunity. But of course, the immune response might not be the same in everybody. So the old individuals and vulnerable people might need to get a booster dose, especially for variants. Now, this is already done for influenza, where susceptible individuals are given the flu shot every year just before the winter for the new influenza variants that have emerged that year. So there is a possibility, a very high likelihood of the same happening for SARS-CoV-2. So thank you. I would like to be most happy to take any questions.
Thank you very much, Professor Nilika, for this informative presentation and a very useful presentation. Now, there are few questions from the audience. I think already you have answered a few of them. Shall I direct you another question?
Yes, yes.
This is another question from a participant. For how many days an infected person should wait for vaccination after viral infection? I think he's asking whether we should get vaccination if you are infected.
Yes. So the WHO and all the authorities do recommend vaccination even if you have had COVID. True that after you have had COVID, you will not get infected for like 3 to 4 months. You are, even with Delta or any variant, which is very unlikely, after getting COVID, that you will be infected with whatever for 3 to 4 months. But after that, the immunity declines, the antibody levels decline, not just antibody, the immune response declines. So then you are not protected. So this is why it is important to get the vaccine. So, and some countries, of course, recommend both vaccines, both doses of a vaccine, or if it's just a single dose, to take that vaccine. Whereas certain countries like France only recommend one vaccine for infected people. And actually, our data also shows with AstraZeneca, Sinopharm, and so on, if you give the vaccine to infected people, then their immune response just shoots, really increases. So I don't know whether one dose might be enough for infected people, but definitely infected people should get vaccinated. And the recommendation is to wait for 4 weeks before getting the vaccine.
Okay, thank you very much for your answer. Then there is another question. What is the antibody level one should have to avoid severe disease? I think,
yes.
Yeah.
Yeah. So again, now it is not antibodies, but it's just it's neutralizing antibodies that are important. Because as I just showed, any antibodies against other parts of the virus are not going to help you in preventing infection. So for the neutralizing antibodies, nobody knows what that level should be. Studies have shown that the more neutralizing antibodies you have, the more protected you are. But people still haven't come out with a cutoff value.
There is another question. Why is the vaccination of 18 to 30 years still not commenced?
Yes, so that is because, of course, everywhere in the world, there's a shortage of vaccines. And that is, which in Sri Lanka, we can see as well. So I just showed you, and I just told you that the more people who are dying are the people over 60s. So then, given what is happening everywhere in the world, and it's the people who are over 60s who are developing severe disease. So it makes sense to stop people dying first, because we don't want our grandparents, parents dying, and they are the people who are going to die when they get COVID. So you have to protect the people who are going to die first. So everywhere in the world, first you give the vaccine to over 60s. Then when you get more vaccines, you give it to under 60s, between the age of 30 to 60, people who have diabetes and other comorbidities. When you have given to those people, then you give to from 30 to 60, and then you can move down to give from 18 to 30, and so depending on the availability of vaccines. So Sri Lanka will move to that point from 18 to 30, because even if you look at the death data, people between 18 to 30 years haven't died. I mean, the very few people who have died are people who have had cancers or chronic kidney disease, or, like very sick people. So people in their 20s and in their teenagers, actually, they do not develop severe disease. This is what is getting the data from all countries. So you protect the people who die and develop severe disease first.
Okay, thank you again. There is another question. How different variants suddenly go out of control when completely controlled in case of Wuhan? In the case, what is the place of vaccine? Is that clear? The question is, now they're asking if vaccines are effective, why you can't control it?
Yes. So vaccines are effective in preventing severe disease and death. Okay, so because I just showed you Pfizer and AstraZeneca, the efficacy in preventing symptomatic infection for a variant like Delta is like 70%, 75%, something, or between 60 to 70%. So which means fully vaccinated individuals, about 30 to 35% of fully vaccinated individuals will develop symptoms, but they are not going to die or develop severe disease. So what the vaccines do is actually preventing severe disease and death. They don't prevent infection. So because vaccines don't prevent infections, the vaccines cannot prevent transmission.
I think it is a very good answer. Again, there is another question. How is the variant predicted from just the RT-PCR? I think this is due to misunderstanding. How is the variant predicted from just the RT-PCR without sequencing? I think I know you
yes.
all are doing sequencing to detect.
Yeah. So you can't predict a variant by RT-PCR. That is impossible. So, but the UK data I showed, the S-drop and not S-drop. So what happened with the UK variant or the alpha variant, because of this particular mutation in a PCR kit called the TACPAR kit, the primers for the S gene were not amplified because of that mutation. So in that PCR kit, because the primers were targeting that particular region of the virus, in that kit, it's a 3-gene kit. The other 2 genes are amplified, but that gene is not amplified. So then everywhere in the world, when you use that kit, if the S gene is not amplified, you know that is most likely to be alpha. And that's how, and when you know that it's alpha everywhere, then you know that if you get S-drop, that is the alpha. Now coming to delta, then with that kit, if you are not getting S-drop, then you know it's not alpha, but that doesn't mean that it's delta. So then you have to, of course, sequence. So sequencing is the only way you can find out the variant. But apart from that, there are other variant detection kits. So I'll explain what those are. So single nuclear polymorphisms. So if you take delta and you compare it with the original SARS-CoV-2 variant, the delta has certain mutations in the spike protein. So in other words, if you want to call a virus delta, the delta has certain unique mutations in the spike protein. So then you have single nuclear polymorphisms of this spike protein, and then you can do real-time PCR targeting these. So it's a SNP analysis. So you do these things to, because you have this mutation. So XY said about 4 mutations that define delta. So when you run a variant PCR, when you detect those mutations, then you sort of get an idea. This is most likely to be delta, and then of course you can sequence and confirm. But given a situation like with your sequencing results, if you find that everything is like 90% is delta, and when you know that alpha doesn't have those mutations, then you can, with good confidence, you can say that with the variant PCR, not a normal real-time PCR, but these are single nuclear polymorphism detecting real-time PCRs, you can say that these are the mutations in this virus, so it is most likely to be Delta.
I think answer is very clear, right? If you want to get a new variant correctly, then you have to do the sequencing. But real RT-PCR, real-time PCR is only one application. The RT-PCR that we are going to use in the detection of COVID is totally different from the RT-PCR that you are going to detect variants. That is single nucleotide polymorphism detection. Okay, then thank you very much for your answer. Then the next question, if an individual got positive for the COVID after he got his first dose of vaccine, what will happen?
I mean, it even depends on what vaccine you had, of course. So, but most vaccines after a single dose does still protect, and you are unlikely to get severe disease. So after you get better, 4 weeks later, you can take the second dose. So that those are the recommendations.
Yes, another very important question. What do you believe regarding COVID spreading among children and need to vaccinate children?
Yes, so that is a question that a lot of people are talking about globally. Now in USA, they are vaccinating teenagers, which are children between 12 to 18. There are no clinical trials in vaccinating children less than 12 years. When we say children, we are talking about 12 to 18-year-olds. And if we can talk about Sri Lankan data about, I don't know, like large number of children, about 50% or something, 50,000 or something children have got PCR positive for the virus. And I believe 12 children have died. And of those 12 children, they have had undiagnosed diabetes, chronic kidney disease, cancer. So it's unfortunate that such individuals, again, we know the risk factors of dying of COVID, and whether you're a child or an adult, if you have chronic kidney disease, cancer, leukemia, if you get COVID, then of course the situation is going to be bad. Now, whether you should vaccinate children or not, in the UK, they are still waiting for more data because whether you have to give the same dose as adults, because a rare complication that does occur in children is myocarditis. In other words, inflammation of the heart. After these, because it's only Moderna and Pfizer that have been approved for children in USA. And so it's not a very high complication, but about 180 children per 1 million. So get this complication of myocarditis with the vaccines. So, it is after the second dose that they get it. So and children have a really massive immune response to these vaccines, unlike adults. So the discussions are right now whether the children actually need both doses. Maybe only one dose is enough for children because they respond much better to vaccines, and or whether you give half dose to children instead of a full dose. Because we know, like other vaccines that are given to adults and children, like you don't give the same vaccine to adults and children because children's immune response is very much different to adults. I mean, it's not the same because they have got a lot of naive immune response. Their immune response system is so good. I mean, it's very robust. They respond very fast. So maybe like even the chickenpox vaccine that is given for herpes zoster versus the chickenpox vaccine given for children, in the vaccine given to 60-year-olds, it contains 10 times more viral particles than the ones given to children. So whether the same one should be given to children is under discussion because we want to make our children safe, but we want to make them safe in a good way. Thank you.
Okay, there are many questions, but I will select, I will try to summarize them. There are many questions on if you take first dose from one vaccine and second dose from another vaccine, what do you recommend?
Yes, so these mix and match vaccines have been done for AstraZeneca, Pfizer, and Moderna, where the first dose is given AstraZeneca, Pfizer, Moderna, and the second dose is something else. So AstraZeneca with these mRNA vaccines have been mixed, and what they have shown that there are no efficacy trials to see the efficacy of mix and match, but what the immunogenicity trials have shown that when you mix and match vaccines, the AstraZeneca with the mRNA vaccines, the antibody response and the T-cell response is so much stronger than using either vaccine alone. So that those are just immunogenicity data. There are no efficacy trials, and there are no other mix and match data for any other vaccines apart from AstraZeneca with Moderna and Pfizer.
There is another question regarding post-COVID syndrome. I don't know. His question is, yeah, what can be done with patients having persisting symptoms of COVID-19?
Yes. So post-COVID syndrome, it is more in people who get severe disease. Especially tiredness, then this difficulty in concentration, difficulty in sleeping, and you might get, of course, not being able to smell and taste for some time. But it's mostly the tiredness and this mental clouding that people get, just being so tired. And this can persist for like 5 to 6 weeks, but it can even persist longer. And this is depending on how bad people were when they had COVID. So studies have shown that actually giving the vaccine to infected people improves their long COVID symptoms. And also when you give out the vaccine and after people develop COVID after getting vaccine, because they didn't develop severe disease, the chances of them getting long COVID is very much less. If there are people who are having long COVID right now, it's symptomatic treatment. Depending on what symptoms the person has, treatment will be done accordingly.
I think there is another useful question. A non-person has taken medicine for food allergy last year, Desdolorine and Montelukast. Now the question is it safe for him to get vaccine?
So this is a really important question because people don't get vaccinated because of various beliefs, and this is why people are dying, especially of the 94 deaths that happened 2 days ago, 79% of individuals were not vaccinated. Okay, so that is sad. So if they were vaccinated, maybe things would be different. So allergy is completely safe for people with food allergies, insect allergies, asthma, allergic rhinitis to get any COVID vaccine. Okay, so just because anybody has a food allergy or insect allergy or asthma or allergic rhinitis, it is absolutely safe for them to get any COVID vaccine.
Yes, some of them asking about the third dose because some countries now giving third dose, like Israel. Now
yes.
There are a few questions about third dose.
Yes. So I think before we talk about the third dose, it is really important to fully vaccinate our adults, isn't it? Because like so many adults are not vaccinated. So I know. So without actually vaccinating all our adults, I don't think we can talk about the third dose. After we finish vaccinating all our adults, then maybe we can think about giving a third dose, starting from the people who are most vulnerable. It is the older people who are going to lose their immune response soon. It is the older people who have other comorbidities, and I just showed you the vaccines reduce the risk of death in older people. So an 80-year-old will have the same risk as an unvaccinated 50-year-old. So it is those people who would need a booster dose after we have adequately vaccinated everybody. And so for booster doses in countries like ours, because we are not US, we are not UK, so the strategy should be to prevent death. It is really unfortunate that people would die, and we should do everything possible to stop the deaths. And to stop the deaths, we have to vaccinate the vulnerable.
One person is asking about any findings on the research on serum testing of infected patients as immune response.
Yeah. So we have our studies are published also. We have looked at the immune responses to all these variants, comparing the immune responses to variants with vaccination, the IgG responses, the IgA responses, the immune responses to various SARS-CoV-2 proteins, neutralizing antibodies at different time points after infection. All that. One is published, the other one is under review, but the preprint is there.
Now there is another question. Is it necessary to get vaccinated people having asthma or lung diseases?
Yes, I mean, asthma is not unlike other respiratory infections like influenza, asthma is not a allergic asthma. Allergic asthma is not a risk factor for severe COVID. But then there are people with other lung infections like chronic obstructive pulmonary disease, emphysema, I mean, so many other things. They should definitely get vaccinated. There's no question about it.
There is one other important question. I think currently a lot of messages are being circulating regarding home remedies. What is your opinion on that?
Yeah, so I have no idea about home remedies. Now, things like vitamin D are likely to have an effect. So now that doesn't mean we know that vitamin D is a lipid soluble vitamin and not a water soluble vitamin. So if you overdose yourself, you will overdose yourself. It will not be gone in the urine. So it is good to take the recommended doses for the recommended time period. Things like vitamin D. You can take vitamin C also, because if you take excess, you will just pass it in urine, so it's not dangerous. But and of course, it is there is no harm done even if we don't know, like things like local treatment like Guru and things. We always take those for respiratory illnesses. So I think that is a good idea to take Guru Apart from that, I have no other ideas about any other home remedies.
This is the last question, madam. If one person having penicillin allergy, do you recommend Sinopharm vaccine?
Yes, if it is only penicillin allergy, Sinopharm vaccine is completely okay, because actually Sinopharm vaccine is an inactivated vaccine, and it is one of the vaccines with the least side effects. So is AstraZeneca. So it's Pfizer and Moderna which causes most allergies in people who have allergies. So anaphylaxis has been mostly reported with Pfizer and Moderna, but Sinopharm is safe.
Okay, thank you very much, Professor Nilika. Now you have spent more time on questions. Anyway, thank you very much for being with us. All the participants, thank you very much. Now, there will be another webinar sessions organized by Young Scientist Forum, National Science and Technology Commission. Other thing is, since you are young scientists in Sri Lanka, if you are not still a member of Young Scientist Forum, you can apply for your membership. You can visit the NASTIC website and download your application and send it to Young Scientist Forum. Okay, thank you very much, Professor Nilika, for your very informative and valuable presentation.